Assalamu alaikum, good afternoon, and welcome to tonight's episode of Talking Point. I'm your host, Syed Niaz Ahmed. In our studios today, we have a very distinguished guest. It's our luck that he's on a short visit to London, but we have been able to convince him that it is important to come to us and also to discuss his achievements, which are numerous, but we are going to unfold things slowly and gradually. Welcome to the show, sir. How are you?
Thank you very much. Good to be here. Yeah.
Nice. What brings you to London?
I came to London to do a program with our local community people. I came last year and engaged with Bangladeshi diaspora in London. And today there's going to be another program to sort of discuss my work and what to do about it in the context of development in Bangladesh and particularly in Sylhet. Yeah.
Well, you are a Bangladeshi geneticist and also a science writer, and you live in Australia. You were born in Kanihati, Hajipur Union, Kulora, Molibuzar district, and then you went to Dhaka University. You got your PhD from Oregon State. Then you went to work at NIH, National Institutes of Health in the USA, and you also worked at MIT, the famous Massachusetts Institute of Technology. It's a long journey, isn't it?
Yes.
Did you realize that you are going to be a scientist and you're going to break through some of the barriers that are natural, that appear natural, but you have been able to break through?
I did not originally, when I was in Bangladesh, I didn't have any concept of becoming a scientist or anything. But as soon as I went to the US at the age of 23, I came in contact with some very famous scientists. And in their presence, I also was able to make some important discoveries very quickly. Beginner's luck, they called it. And that made it possible for me to think to be like those people.
Right.
So at the age of 24, I was already making some discoveries.
What was that discovery?
Initially, it has to do with what people call CRISPR-Cas9 or gene editing. We used to call it recombination. There was a different language at that point. Gene targeting or recombination. Simply put, it is the idea of making some specific changes in the genome. So make a crop which does not yielding a lot. Now you want the crop to yield more. So you can make targeted changes in the genome. You can kind of rewrite the genetic code, if you will. And the technology is called genetic recombination or gene editing. Now they call it CRISPR-Cas9. So I made some fundamental discovery of this molecular machine which opens up the DNA so that you can make changes in the DNA. That was my work in PhD.
Was it a calculated discovery, or was it just a by chance you did come along?
It was not a calculated discovery. My PhD supervisor said, this is really the field. Now think about it and see you can do something about it. So this is basically how a PhD student is told to kind of go ahead and make it.
You opened the closed doors.
Yeah, make your mark. I mean, this is the American system that you don't tell people what to do, but let them figure out what they're going to do. So that really suited me very well. So
See, this together with colleagues in Australia, you isolated FIS, which is fertilization independent seed. These are the genes, and FIS mutants can produce partial seed without fertilization. Like producing things without a quote-unquote father.
Yes, of course.
That is remarkable.
Yeah, so that was my, other than the recombination work that I just described, that was kind of my next major work. And I did that in Australia. So I like to say that I did something big for America when I was very young. Then I moved on to Australia in my 30s. And I did something big for Australia as well, which is fertilization independent seed or seed without father, or some people call it virgin birth mutant. I kind of conceive, well, the context always is there. Nothing comes from a vacuum. So Yeah.
Immaculate conception.
Immaculate, yeah, immaculate. I used to say immaculate conception, and somebody corrected me and said, no, it is virgin birth, which is not immaculate. It's just technical discussion around Christianity. But virgin birth is an idea that obviously, as you know, comes outside science. But in science, there is a similar concept called apomixis. Apomixis is the process by which seed is formed without fertilization.
Right.
But nobody knew how it happens. So my discovery is to do it from the scratch, so to speak, to actually create it in a targeted way from nothing. This is what my discovery was. So I found that you can do it. We found 3 genes, FIS1, FIS2, FIS3. And when you mutate them, then the seed starts to form without any pollination. And recently, in a cover story in Nature Plants, Nature magazine, we did it in rice. Me and my colleagues, mostly from China.
Right.
It took us about 30 years to do it in rice, but we did it.
When you say we did it on rice, you see, so that means we can do it with other fruits and plants?
Well, initially we did it in what they call model plant. It's like a mustard family plant called Arabidopsis. This is something I did in the 90s when I conceived this experiment and I executed it. And those papers were published in 96, 97, around that time, 1996. But then there was a long hiatus when I was given a lot of money to do it in rice, but I couldn't do it in rice. But we kept on doing it, and in the background I had a large group of colleagues, mainly in China, and they were plugging away at it. And my other colleagues in Australia, particularly Ming Luo, I want to mention his name. So mainly because of the effort that Ming was continuing, even in my absence, we finally could do it in rice, the so-called virgin birth mutant in rice. We had to knock out 2 different genes by a process called CRISPR-Cas9, where you go in and mutate individual genes. So we did that, and then after mutating 2 genes, it's now happening in rice. And that was published on the cover of Nature.
Is it that you confuse the plant?
No, you don't confuse it. You basically understand the fertilization process in detail. You separate out the individual component that is involved in fertilization process, namely the paternal father's contribution, the mother's contribution. You learn it in great detail. And then you learn to intercept or create mutation in a way that the father's contribution is redundant, is not needed anymore. So this is really the strategy you take. Yeah.
Can it be extended to, as I asked before, so can it be extended to other plants, fruits, and by God, can it be extended to human beings?
Well, let's talk about plants first. So it can be done in plants. So I can see an obvious way to do it in maize, in corn. So I'm taking steps to do that, and wheat, etc. Or just about any crop it can be done. And then in mice, one of my colleagues, Azim Surani of Oxford, many years ago, he showed that it could be done in mice. It's called parthenogenetic. It's also called parthenogenesis, where the egg or the sexual cell, as we call, becomes the embryo. So in theory, it could be done in any living organism, but in a case-by-case basis, you have to do knock out specific genes or add other genes. So it's a genetic apparatus, you know, and if you understand it fully, you can intercept and make things happen. For obvious moral reason, you're not going to attempt it to do it in human beings.
True.
Yeah.
Are others working on this kind of project?
Yes, so when I isolated the FIS mutant, we were the first to do it. I did it in Australia with my colleagues in CSIR, or plant industry. It led to a new field called the FIS biology, fertilizer independent seed biology. And then obviously, because it's so exciting, other people got in. So there is a FIS biology community that sprang up basically on
all over the world.
On the basis of my work, it sprang all over the world. And recently I was invited to go to Argentina to talk about the 30 years of FIS biology in a town called Rosario, which happens to be the city where Messi is from. So I had great time flying all the way to Argentina. It's so far away from Australia, I felt like going to outer space or something, but I was there and I spoke in that conference in Argentina. So it was a great feeling to see that my work, which started from a random thought in 1993.
Are the Argentinians also like us rice eaters?
In Argentina, you mean? Yeah, there's a lot of rice in Argentina as well, but mostly wheat, yeah.
So have you tried this on wheat?
I have not done it in wheat. Everything is a separate project involving a lot of money and things like that, but there is no reason why it can't be done in wheat, yeah.
It's a question that's been, since I learned about it, it's been, we talk about overpopulation, we talk about the scarcity of food, and now when I saw this, I said, oh my God, this is the answer, this is the solution, eureka, we have found the final. So will it, in future, will it totally solve the food problem?
Well,
it should. Theoretically.
It will make long strides, because one of the things that you can do when you put apomixis into the crop system, you can make hybrid vigor perpetuated forever. Because normally when you make a hybrid plant, farmer grows the hybrid plant and the seed they get from it cannot be saved because it doesn't have the hybrid vigor anymore. It breaks down. But if you put in apomixis, or the virgin birth or the extended virgin birth technology where pollen is not needed, but the seed are kind of a clonal, we call it clonal copy of the mother delivered by a seed. When you do have that in a cropping system, then you have the hybrid vigor perpetuated forever. So this is one of the reasons why apomixis is so attractive in agriculture. And
so this Boro thing, is it that the same plant will produce 5 times of the crop?
So that's another thing I have done. So one is the apomixis, which is virgin birth, and what we call perpetuation of hybrid vigor or heterosis is one work that I have done. And the more recent work, what you are referring to, is that same plant gives harvest 5 times.
Yep.
So it has some similarity with apomixis.
So is it one after the other, day after?
One after another. So it has some similarity in the sense that you have a plant, but you're not going to get only one harvest from it. You will get repeatedly 5 harvests. So whatever vigor, let's say if you were to make a hybrid, that hybrid will give 5 crops in a year.
One after the other?
One after the other, yeah. So that's another mega project.
So the weather does not affect?
Well, weather can be very extreme and it will affect. So I cannot say that weather will not affect. But this is a separate project where I conceived that why wouldn't a rice plant in this case give many harvests?
We can have giant green sheds or something that
Yeah, the problem is that like in Bangladesh or India or most of the tropics, there's a big problem where you harvest and then you have to dig up the soil and you have to put the seedling again and get the aush. So you start with burro and then aush and then amon. So it's very tedious. I actually come from a village, and my family historically we were agricultural landowners, so we were very tied to agricultural system. So I have seen how it is from the very early days of my childhood. And if you can have a crop which gives you multiple harvests after the same planting, it's a great thing, you know.
So you do not know any other crops, so you just one crop and gives you 5 times of the products.
Yes, yes.
So what was that when I was a kid? I heard about something like Erie, and they were talking about it that it doubles or triples the product.
This is something you are referring to, what is known as Green Revolution, where IRI stands for International Rice Research Institute, based in Manila, set up in the 60s. And so in Bangladesh we call it Iridhan. Means this is the modern rice, high-yielding variety, HYV also it's called. And this replaced our traditional varieties. So IRI came along in the 60s with the claim of Green Revolution, and then there was a lot of increase in the yield, and that is considered to be the one that prevented all the famines and so on. So the high-yielding variety or modern rice variety became synonymous with the word iri, and that promised increased yield by a factor.
This one is called BRRI?
BRRI is Bangladesh Rice Research Institute, called BRI, which is kind of iri of Bangladesh.
So this is your contribution to this?
No, I did it. All my work I did it as an individual. BRI is a separate organization which have many varieties in Bangladesh. You must have heard Biridhan 28, Biridhan 29.
Yeah.
So these are landmark mega varieties which is very much in the agricultural system. People are growing it. But what I did is my individual effort together with the farmers of my village.
Right. Your this work, has it been accepted by the different governments, and do they plan to put it into implementation and use it for the benefit of mankind?
Of course. Well, it is in the process. I just did it. I let it be known that I have done it. I did many varieties. I'll make it available to other people who will have to repeat it. There's a scientific method, right? So I share my data. I give the seed to them. I give the method to them, and they repeat it using my method, and then they say, yes, we got it. So this process has just started. I'm just a lone individual doing it with my farmers, right? So I don't have an institution behind me for this work. So it will take time, right? I have done it 3-4 times. We don't say anything before we have done 3-4 times.
Of course.
So now the varieties exist, the method exists. So I'm going around the world, including Bangladesh. I'm saying, look, this is what I've done. Please take the seed and repeat it.
What sort of response did you get from people in Bangladesh? I mean, particularly your own community.
Oh, very good response. As you may have heard, this news became very viral. Everyone is sharing it. It's a feel-good news, right? Because people are tired of bad news in the world. So a rice giving 5 harvests.
Good news.
Pancha, 5. 5 is very close to people's heart.
Yes.
And dhan, brihi, is the old Sanskrit name for rice.
Right.
So pancha brihi is become a catchword, and people are sharing it in social media in particular.
Is it going to be Boro or Aus or what?
It will start with the Boro. So you start in February, and end of April you get the Boro crop. We just got our Boro crop, right? Then you cut it in a particular way, and you take your crop home. But from that remnants of what you have cut, from that debris, from that something that is just a waste normally, a new plant will emerge, right? And then 45 days later you are going to have a second early Aush harvest. When you cut it, it will grow back again, and it will give you a second Aush harvest, and then subsequently it will give you 2 almond harvests about 45 days apart. And by the time the year is ending around November, December, when winter is coming in, you would have gotten 5 harvests from the same planting.
From the same land?
From the same land, from the same crop. You get 5 harvests.
And the farmer doesn't have to till the land again and again.
Farmer doesn't have to till the land again and again. Farmer needs to clean up the debris because there's going to be excess mess. It has to clean up and look after the plant that he has just cut a bit. There is a procedure. We'll supply that procedure. And then this particular variety, which has been constructed to behave like this, this is one thing is very important. A random variety will not behave like this.
Right. So we need to take a break, and then when we come back, we will continue with the same discussion.
Thank you.
Thank you very much for being with us. We need to take a break, and when we're back, we'll pick up the same thread. Thank you. Nice. Welcome back. For those of you who have just joined in, I have a gentleman as my guest today. He is considered one of the most powerful thinkers, writer, and well-known geneticist, Dr. Abid Chaudhry. He is based in Australia and Bangladesh. He is considered one of the most prominent leading figures in biology. You have an organization called Genofacts, you see. What does it do?
Genofacts is something, I'm a co-founder of Genofacts along with another entrepreneur of Bangladeshi origin called Jahangir Alam. We connected in Sydney, and we conceived the idea of Genofacts together. And so Genofacts, if I want to say,
I like the way you use the word connected and conceived.
every new entity has to happen through connection and conception, right? So Genofacts is an idea which falls under the umbrella of what is called digital medicine, as opposed to old-fashioned evidence-based medicine. So what is digital medicine in a very specific, direct way? It is based on DNA analysis. In this particular case, DNA is taken from your stool sample, and DNA is made out of from the sample that comes from the human underbelly or stool or gut, we call it, gut microbiome. And the DNA sequencing takes place to find the identity of the beneficial bacteria that you carry in your body. It turns out that you have more bacterial cells than your own cell. We didn't know about this only 10, 15 years ago, but now we know. So out of the DNA sequencing, we get a complete shopping list of all the bacteria or microbes you carry in your body. And based on their very nature and what they are doing, an artificial intelligence and machine learning program gives an assessment of your current state of health and hopefully your future trajectory of your health and wellness. So this is a very high-powered AI, ML-driven modern medicine, digital medicine, DNA medicine, you can call it. And
so how far have you gone ahead in this?
We have a minimum viable product, which is the gut test, which will take the stool sample, isolate the DNA, sequence the DNA, and give people a report, which is an advisory on the state of their health. We cannot yet give medical advisory because there are regulatory barriers, but we can give wellness advisory, health advisory, lifestyle advisory, suggesting people to alter their lifestyle is needed so they don't get premature diabetes or heart disease and things like that. So it's a very powerful predictive rather than, because normal medicine, you get sick, you go to the doctor and you get a pill.
Right.
This one predicts what is about to happen, what will happen.
Yeah. Well, it's predictive as well as, when it is predictive, it can be preventive.
It's preventative, it's predictive, it is personalized, it is targeted to your own situation, and it is also digital in the sense that it's DNA driven, DNA sequencing driven, and it is also bolstered through the analysis of AI and ML and the supercomputers.
Oh, this is wonderful because you have been involved in the rice production and increasing the productivity of the crops, and now you have gone into something which is there but has not been explored as yet, and this is what you call gut
microbiome.
Gut microbiology, see, and finding what is on the horizon, not what's happened just now, but what's on the horizon, what could be coming for a particular person or his chart for the diseases that he will probably come.
Yeah, and indeed, depending on how many people would take the test for the whole community.
Yes.
So let's say in Bangladesh, people are getting cardiovascular disease, diabetes at a very early age.
Right.
And they are going to the doctor who is giving them medicine. People are developing kidney problems. What the gut test and our Genofacts platform will do, that it will predict what is about to happen. So it's a public health intervention of a statistical and whole population nature, which will which will first of all foreshadow the trajectory of what is coming in this population, and then try to fix it before the calamity has occurred. So
we come to the basics of the thing. You see, people in Bangladesh, particularly, they have a tendency, those who can afford, they have a tendency to go abroad rather than see the local doctors. See, I do not know why, but since I don't live there, so I do not know why. See, but those who can afford, they will always go abroad. See, is it because our own facility is not up to the mark, or they don't trust the investigations, or there's just an excuse to go abroad and have some fun?
Because I don't live in Bangladesh, I cannot make a very detailed opinion about it, but obviously it is a trust issue, like people like to go overseas for whatever reason. We are trying to change it, because if we do a credible analysis of what is about to come when somebody is in their 20s and 30s and so on. And understand. One good thing about this GenoFX type analysis that it creates a complete analysis of the state of the health, and it gives a very precise explanation of what is happening. So I think as more and more people do the gut test, so to speak, then a new situation will emerge where people will take charge of their personalized situation, their own health, if you will, and adjust the lifestyle, the probiotic, we call it, which is the good bacteria. We'll also make that available. And also the choice of food, what they are eating and what they shouldn't be eating. All coming together in a very personalized, predictive way, which I hope will make a big impact in the future.
Right. So we really do not know why people want to leave Dhaka and go to Hong Kong or Singapore, but maybe they have better hospitals.
Yeah, well, they do have good hospitals, but we can do the same thing. I think we have to put our mind and make things happen in Dhaka.
If we can have scientists like Dr. Abid Chaudhry, why not we can have better hospitals?
Inshallah.
Inshallah. There are some questions that have been sent by my viewers to ask you. Yes. What is the difference between DNA and genes? They might appear very basic to you, but out there somebody wants to know.
Of course. So gene is, you can call it a complete recipe, right? If I want to use the metaphor of a recipe, of a cookbook. So a gene is where the whole recipe is written to, let's say, to define your eye color or whether the hair is curly or not. And DNA is the script in which the recipe is written. So DNA is the alphabet, you can imagine. DNA is the alphabet in which that recipe for your hair, for your eye, for your skin, for your lung, everything is dictated by a gene or many, many genes. And the language in which that code is written is DNA.
That's DNA. Thank you. How is that sometimes we inherit certain traits from our parents and sometimes we don't?
You always inherit your DNA from the parents. Whether that translates into an actual visible trait or not is dependent on some complexity. So let's say, for example, if your mother has a particular eye color, a blue eye, and the father has black eye, then you are likely, so the blue is always hide behind black, right? So when what we call the hybrid with containing the blue eye color gene and the black eye color gene, then the carrier of both alleles, the eye color will be black. It is called dominance and recessive. This is a
but it's not the combination of the colors.
It will not get into a hybrid color. It will just be black, because black is dominant over blue, as we call it. But when that child, which carries both blue and black, later have children, then the blueness and the blackness will come out separately. It is called independent assortment. And then again blueness will come back in the next generation. So it's a bit, we call it Mendelian genetics. You have to know this interplay of the gene and how it pans out through generations.
Right, very interesting, very interesting. Can I grow out of a genetic condition?
Yes, you can. Some genetic conditions are very powerfully ingrained or hardware programmed, like thalassemia and things like that. Other genetic condition is subject to the lifestyle or kind of food or what we call epigenetic modifier intervention. So it can be managed, it can be modified, and people can have healthy lifestyle in spite of certain genetic conditions. So you have to look at it on a case-by-case basis.
Okay. What causes Down syndrome?
Down syndrome is caused by chromosome abnormality. So it's a very different kind of genetic condition. It's a whole set of new situation where the chromosome number itself is altered. So that's broadly called Down syndrome. Yeah.
But it's nothing to do with genetics?
It has to do with genetics, but it has to do with the gross alteration of the actual chromosome number.
What can cause that?
It is caused by when the cells divide, then the chromosomes partition in a very precise way. So when there is a problem in that partitioning, that leads to chromosome number abnormality, which is Down syndrome. Yeah.
And what does genetics have to do with cancer?
Genetics have a lot to do with cancer in many ways. When the cells divide in an orderly way, we have a normal cell division. When that cell division becomes out of control, then you have when you have massive proliferation of cell division, that causes cancer. And one of my teachers, Lee Hartwell, actually got Nobel Prize for discovering cell division cycle gene, CDC genes. So all cancer is caused by abnormal cell division, and therefore at some level it is of genetic origin. But it is not a simplistic hereditary. There is no cancer gene as such. Well, except for breast cancer, BRCA1 and 2. But normally cancer happens due to alteration in your regulation of your cell division as a part of your lifestyle. So let's say if you're smoking away, then there's a hit, these smoking substances goes and hits your DNA, and then unproliferated cell division starts in your lung, or could be in your intestine and so on. So these are genetic, but not hereditarily genetic, but genetics of your own cells. We call it somatic cell, your body cells. But obviously it is due to alteration in the genes.
But in this country and all over the world, I think men seem to suffer a lot from prostate cancer. And they say that people from particular race, they suffer more. Is there any particular reason?
I don't know the details about hereditary nature or the population-wise distribution of propensity to prostate cancer. I don't know detail about it, but it's possible. Now people are thinking that a lot of statistical proliferation or statistical increase or decrease in certain population of certain cancer in fact has genetic origin. It's a new field, and they are deploying artificial intelligence, machine learning, supercomputers to scan the whole genome to find a signature of such things so that they can ascribe it to into a genetic origin of certain type of cancer. A good example is that for the breast cancer, they have found the smoking gun, BRCA1, 2, those kind of gene. For other cancer, the tendency to have cancer, whether or not there is a genetic origin, is a open question and being looked at case by case basis.
Thank you. Human beings may look different, but 99.9% of our DNA is the same.
Yes.
Any particular reason?
Yeah, so it's not just that your DNA, of course, our DNA is 99.9% equivalent, is the same. So even out of this similarity, there are differences in whether the gene will be expressed or not. So one is the code, and the second is how much of the code is expressed. And that expression differences often dictates what we call the appearance, like the shape of our nose, the shape of our lips, or eye color, or hair, curly or not, is dictated not only the genetic code, but also the expression of the gene. So this is why it's a very tangled kind of thing.
Here's the last question. See, humans share about 50% of our genes with plants. Is that right?
I don't know about that. Where did you get that one from?
I don't know. Somebody who is learned. Well, and the last question. Genes make only about 3% of your DNA.
Yes, that's very true. Yeah, so most of the human genome does not contain genes, about 30,000, 25 to 30,000 genes. And the rest of it is what is now people call junk DNA or dark matter. And we are grappling, I myself involved in this research, is to make sense of what we call the non-coding region, this vast ocean of DNA there, which is not coding for genes, but obviously there for some reason. So yeah.
Well, behind this very sensitive mind, there's also a very sensitive heart. Our guest today is also a poet. Can you just briefly tell us something about your poetry?
Poetry started coming to me, and now looking back from memory at a very early age. I have been a writer of poetry, but kind of quiet writer. I didn't proclaim myself as a poet or anything. But it is kind of the inspiration of how things take shape in my mind. It comes through poetry, so I kind of explain life and science and everything through poetry. And I also memorized at a very early age. Now I know, even before I was 9, I had memorized like hundreds and hundreds of poems. So this is
any particular poet you liked?
Well, Rabindranath, of course, I like. He's the best one, but obviously other poets also, Kazi Nazrul and English poet T.S. Eliot, and you know, I like in the French poet Baudelaire and Louis Aragon, so many poets, Paul Éluard.
Yep.
I like. I'm very inclusive about the poet Pablo Neruda, who wrote in Spanish. So I like a lot of poets in multiple languages, and I have made it my business to learn as much poetry as possible by others as well as write myself. Yeah.
That reminds me that you have spent some time in Paris as well.
Yes, yes. I spent a lot of time in France. I'm kind of a Francophile. I went to Dhaka Alliance Française at a very early age, picked up France. So I was very obsessed about French poets and deployed a lot of my energy to learning French and French poetry.
Thank you very much, sir, for coming along and giving us some valuable time of yours. You are on such a short visit to London, but inshallah, some sunny day we'll meet again.
Thank you. Thank you much. Yeah.
Thank you very much for being with us, and I hope that you've enjoyed the show as much as we have. It was all in a rush, but we hope we have come up to your expectations. Thank you. Bye for now.